Correct? What is your research work in virology?
Yeah, I want to learn few topics about virology, sir. As far as now, we are working with the bacteria and fungi. So I have even virus work in my project, RUSA project. So I want to test the antiviral properties of the medicinal plants. So I wanted some
Prakash,
input from the
Prakash Halami sir will help you how to
Definitely.
test.
Good morning.
Sir, I had
It
the
sounds to be
opportunity to meet sir. I went to a workshop with you, sir, in IASP. You
that
remember?
I CFT.
Sir,
CFT, I remember you very well.
thank you, sir.
Nice to meet you. I am actually in US now.
Yes,
With
sir.
reference, just to briefly tell the participant that I am associated with this COVID-19 testing. So this is
Yes.
what my expertise is on the virus handling and detection methods. So I will be definitely, like the other members actually can participate and interact with that. No?
Yes.
Otherwise,
Yes.
also my address is there. Yes.
Okay. Thank you.
Thank you.
Thank you.
Again, Neumann Putra is there. Neumann Putra from which place? Neumann Putra.
Yeah. Good night, everyone. I'm from Udayana, Bali.
Correct. That's why I called you. You are out of India, but you have participated. Welcome you.
Yeah. Thank you.
Yes.
Thank you.
So, can you please tell in short that on which topic of virology you are working in Bali?
Yeah. Actually, I'm just start to working in oncogenic virus.
Okay. Okay.
Like HPP and EBP. Yeah.
Very good.
For HPP, I already work in with the obgyn and gynecologies, and also last time I worked to detect HPP in HIV positive pregnant woman.
Very good. Very good. So again, I will request everyone to write down your name, address, email ID, and contact number. We want to prepare the list of all research worker interested in virology. Okay. So it's a very good Putra, sir, and welcome you. Now, Prakash Halami sir and Biprana sir, please. So first of all, Prakash Halami sir, in short, you tell the importance of virology research.
Yeah, exactly. We are actually looking the antiviral activity of the several probiotics, including the antimicrobial peptides also. And we see that the several probiotics has the antiviral property with reference to inhibiting their replication, with reference to inhibiting their protein synthesis also. So that is a one part. You know that in a food also there are several viruses, as a foodborne viruses, it comes in a picture. So we are actually looking how this can be actually prevented through the food molecules. So this is the one part. Other area which I was actually associated with detection of the COVID virus using RT-PCR, which everyone knows, and MBSI has contributed enormously. And Dr. Deshmukh sir knows very well that MBSI even was associated with honoring our COVID warriors in CFTRI. The people, those who were associated with testing the samples. So through MBSI, we have actually organized such a activity honoring the people, those who were associated. And in this mission, we actually tested almost 7 lakhs COVID samples, for there by using RT-PCR test. Yes. So now everyone knows it became very common, and I believe that RT-PCR again started in India testing for this COVID virus. Microbiology in general and particularly the MBSI activities has a lot of importance in the days to come. Thank you, sir.
Thank you so much, sir. Hello, am I audible to all of you? Hello.
Yes, yes, we can hear you.
Yeah.
Hello. Yes,
So
sir.
good evening, everyone. So my research field is medical microbiology. Mainly, I work on the antibiotic resistance. But now I am shifting to the new area that is alternative to antibiotic. So I am searching for the quorum sensing inhibitors, both from the synthesized chemicals, compounds, and also from the phytocompound, means from the natural plant source. So I have written two reviews on the alternative to antibiotics, and I've gone through the new term for me, that is the phage therapy. And phage therapy, the emerging, you can say that for the alternative to antibiotic, and especially against the multi-drug resistance bacteria. So I take this opportunity, means in this form, to if anyone is expertise in the phage therapy, so I would like to collaborate with any person who is expert in the phage therapy or working on the phage therapy. And also I want to work on the environmental surveillance of viruses, means mainly in the wastewater treatment, means the public health monitoring. So this virology is the new field for me, but I would like to collaborate with the people who are working in the both field, means alternative to antibiotic, mainly the phage therapy, and the virology in field of the wastewater treatment. Thank you very much.
Okay, okay. Anyone working in virology?
Yes, mainly in the two fields, the phage therapy and the wastewater treatment by helping.
No, I'm talking about
Yes.
any other teacher now working in virology. Please come forward and tell in short.
Yes. Am I audible?
Yes.
Yeah, but
Okay. So I'm Dr. Bibhudatta Mishra. So currently I'm working as an assistant professor in NIST University. So I worked on viruses previously, and currently also I'm working on viruses. So in my PhD work on flaviviruses and dengue. So all computational work, docking and simulations. So hello. Yeah.
you're audible, sir.
So in my postdoc from AIMS New Delhi, so I was working on SARS coronavirus 2. So it was a collaborative project between THISTI and New Delhi, SARS coronavirus 2. So all that pseudovirus production and entry assay and all that. So currently I am working on chikungunya virus. So my PhD student is working on chikungunya virus. So in the chikungunya virus, we are looking for three things. So we are working on the RNA secondary structures. Apart from that, we are working on the protein-protein interactions. Apart from that, I am working on the protein ligand interactions, that protein and small molecule docking and simulations. Those kind of thing I am working right now.
Very good, Vibhudatta sir. I will request you.
Yes.
Bipran sir is a organizer today, convener today.
Okay, sir.
You give all your information to Bipranas. Why? Many people in India are in search of the speaker working in virology. I will forward your name.
Okay, sir. So,
Okay.
so can I get the email ID or a phone number of Piprina
Yeah,
sir?
Bipran
Yes, I
sir,
will.
please type. Bipran sir,
Sure, sir.
please type in the chat.
So actually, I was trained in both computational as well as molecular virologist.
Very good, very good. Bipran sir will type his email ID and contact number also, WhatsApp number, so that all are requested to give their contacts, affiliation, everything, if they are having experience, to Bipran sir. Now, Bipran sir, please take the charge to the next speaker.
Sure, sir.
Any other speaker? Any other person working in virology? Hello?
Shared my email ID and WhatsApp number
Yeah.
in the chat box.
Yes. Any other person? Any other attendee worked on virology? If not, I will request. Sorry.
So I am sharing some topics related to the virology. If anyone has
Okay.
experience, so just share your experience on this topic.
Correct.
I shared with
Sai,
the
Sai Teja is there. Sai Teja, can you focus on virology MSc?
Yes, sir.
Yeah. So you tell some information of the MSc virology in Venkateshwara University.
Yes, sir. We recently completed MSc Virology from Virology University, where we learned about recombinant DNA technology and the plant virology and animal and human virology. Sir, am I audible, sir?
yes, you are audible.
Yes.
Yeah, we generally some theoretical knowledge and basics we have learned it. Yeah, and we have gone through a recent training program in FABA, that is Federation of Asian Biotech Association. We have learned some foundations in cell biology, microbiology, and molecular biology. Presently, trying to pursue a PhD. That's it, sir. Hello, sir. Sir, am I audible?
Yes, you are audible.
Presently,
You're audible.
trying to complete a crack the CSR, ICMR net to pursue PhD, sir.
Yes.
Yes.
Okay. Sir, one of the fellow from Bali also has joined.
Okay.
Yes. And today, Prakash Alami has joined from USA.
So it's great, sir.
Yes. So, Khan Arbas, he is from Government Institute of Science. So, whether you have worked somewhere in virology, Dr. Sandeep Yadav is there, Gurgaon. Sandeep Yadav, please.
Hello. Hello, I am audible, sir.
Yes, you are
Yes.
audible, Sandeep.
Hello, everyone. Good evening. Myself, Dr. Sandeep Yadav, and I am assistant in School of Life Sciences, Starix University, Gurugram. And currently, I am working on the viral genomics. Basically, we we are targeting a specific type of repeat pattern. We develop a technique, basically a manual bioinformatic based technique, in which is usually uh, identify a specific repeat pattern named as mirror repeat and in the viral genomes. So currently we are doing the viral repeat based study, and in my previous research is also by using this same method on different kind of domain viruses like plant, animal, and even in on coronavirus also. And we currently my dissertation student, MSc students are also doing on fungal based research by using the same approach. Hello,
Yes, sir.
I am audible, sir.
Yes, sir, you are audible, sir.
So we are basically I my field of specialization in the viral genomics, and we developed a unique type of methodology for the specific repeat type, and we just focus on that repeat pattern. But it's only a bioinformatics based analysis, not a experiment. We are currently going for the experimental analysis of these sequence also, but our study is basically focused on the identification of these sequences in the viral genomes. And our previous studies find that these mirror sequences are actually thoroughly distributed in the all kinds of plant, and they are showing unique kind of conservative nature in every family of the virus. For example, like in case of HIV, we can see 231. We reported 231 mirror sequence, which are only unique to that HIV domain only, HIV viral family only. They are existed in the HIV only. Not they are not found in the other viruses. So these kind of study we are doing currently. Thank you so much.
Thank you.
Yes, one more madam is there. Vancha. Hello. There is a Vancha Madhu. Mr. Vancha Madhu. Mr. Vancha Madhu, can you talk something? Mr. Vancha is assistant professor. If not, Biprana sir, go ahead.
Okay. Bhavana madam is also joined. So Sampada Ashok Patang is student of MSc Microbiology. Do you want to share some idea with us? Sampada Ashok Patange.
हेलो।
हाँ,
Yeah,
yes.
audible.
हाँ सर
Very
बोलिए।
good.
हाँ
हाँ, आप कहाँ एमएससी करते हो?
गवर्नमेंट मेरी अभी कंप्लीट हुई है सर। गवर्नमेंट इंस्टीट्यूट में कर रही थी मैं, गवर्नमेंट इंस्टीट्यूट ऑफ साइंस छत्रपति संभाजी नगर।
हाँ, ठीक है। तो वायरोलॉजी का कुछ काम किया था क्या एमएससी में?
प्रैक्टिकल किया था सर।
ओके ओके। अभी कुछ रिसर्च के सर्च में हो क्या?
नहीं नहीं सर, अभी फिलहाल मतलब मेरी अभी एमएससी कंप्लीट हुई है, अभी मैं फिलहाल घर पे हूँ।
ओके ओके। सो एनी otherwise I will request any other student or any other teacher want to focus the light. So there are many, about 30 are there. Rukaya want to talk? Otherwise, again I will request Vibhu Datta, Prakash sir, Muni Kumari, to Bhavna Pandey. Pandey can speak. Okay. I will request Prasun Kumar. Prasun. Hello.
Yes, good evening, sir. Good evening, all of you.
Good evening.
Yes, sir. Actually, my background is not virology, so I am not speaking anything. I am working currently. I am working as an assistant professor at Sharda University, and my area of research is related to bioconversion of waste to value-added products. And I am
Okay.
sidewise also working on antibiofilm activities of natural compounds. So
Very
it is
good.
more of antimicrobial things or antibiotic things, but not on viruses actually.
So I will request again Prakash Arami sir.
But for the sir, yes, for the topic of discussion, the recent discovery that came into Science magazine, using CRISPR scientists have shown that they can remove HIV permanently from the cells. So that is a hot news nowadays in the virology sector. I thought somebody would be discussing this fact. Yes,
Okay. Because CRISPR technology is now widely used.
yes,
And
sir.
you are talking about the use of CRISPR technology in virology.
Yes, they have removed HIV virus permanently from the cells. So it is
From
quite
the
a good breakthrough.
HIV from?
From cell line, sir.
Okay.
Eukaryotic cells. Yes.
Okay. Sir, Prakash sir
Yes,
can
sir.
focus the line.
Yes, sir. What
Uh,
the previous colleague was telling, participant was telling, almost on the similar line. The adjacent department here work is going on, where the CRISPR they are using to inactivate the virus, HIV virus. I am presently at the University of Commonwealth University of Virginia Commonwealth University in a biomedical health center. And here one department actually is exclusively working how the HIV virus can be inactivated using the CRISPR technology, very rightly told by the previous participants. And on the similar line, the several research activities are also going on using either the using the CRISPR tool, and then several other methodologies where the HIV has been now found to be much more either at the detection level using the PCR or RT-PCR methods, or making sure that the virus antibodies are generated, or even the making the virus totally inactive. You know, on this area also the work is going on. As I pointed out initially, beside these human viruses, we look the zoonotic viruses several. You know, and my colleague at the Bangalore, what we have the institute there, the diseases are actually the cause of the diseases associated with the human being also, you know, and that's why the zoonotic virus also is a major cause of concern. And if we see the history even of the COVID virus, this is basically again comes from the animal sources, right? And looking this, because most of these animals actually act as a natural reservoir for most of those viruses. And as you know, that virus genome actually is much more dynamic, goes on changing. So developing only the detection tool is may not be the enough, right? Looking their surveillance, ensuring that there are different virulence, serotypes, what being developed is also important to have such surveillance on these viruses. And from that angle, what microbiome MBSI is looking for a very combining the discussion from the researcher from the different field. It was a very healthy discussion what we have seen from last 1 hour, half an hour, on this background. So definitely connecting with each other, the people from the academic, the people from the research institute, the universities, including the companies, several our, the vaccine which was developed actually by Bharat Biotech was quite useful, right? Covaxin, COVID vaccines, So such information are the research institute, National Institute of Virology, Pune, or NARI, National AIDS Research Institute, again in Pune. Most of you people are then several viral stocks are actually available, plant viruses, in IRI, New Delhi, you know. So having this information from this national facilities as well as the National Institute is another actually requirement. One of the colleague what he was referring was even the information pertaining to mm, um, this looking the viruses for the purpose of what did you say? You know, no, how we can actually make this virus as a delivery system. So several aspects are there. Definitely, I think we are in a very right track to contribute for the society through this MBSI platform. Thank you, sir.
Very good. Very nicely illustrated.
Yes, sir.
Now, anyone again want to speak something about the virology?
Sir, in my opinion, sir, Dr. Sandeep Jadhav and Bibhu Datto Mishra sir can discuss on the topic because they are working on the virology.
Very good.
Right.
Bibhu Datto Mishra sir.
Ashwini want to speak something.
Okay.
Ashwini? Hello? Whether Ashwini want to speak something? Otherwise, Bibhu Datta will proceed.
Okay.
Ashwini? No, Ashwini don't want to talk. Bibhu Datta, you can proceed.
Should I continue or?
Sure, sir.
Yes.
Okay. So can I tell something about my PhD and postdoc research or currently
Correct.
research?
Correct.
Okay.
You can tell.
Okay. So I said, in my PhD, I was working on dengue and other flaviviruses. So in that, what we were checking, like whether any dengue is actually of divided into serotype, dengue 1, 2, 3, and 4. And again, these serotypes are divided into different genotypes. Like, for example, dengue 2 is divided into different genotypes like Asian 1, Asian 2, Asian American, American Cosmopolitan. These are the different genotypes of dengue. So what we are doing that, what I am doing in my PhD. So we are checking, is there any sero and genotypic differences are present in that, and we are correlating that differences into the onto the structural level. Means sequence level, lot of works have been done, but in the structural level there is not much work. So what we are doing, we are just correlating that what are the structural differences which make a impact of these differences. So that was my one of my objective. Apart from that, we are also interested to study, like we are also seeing what are the different secondary structures that is forming. So what we found, like in case of dengue, is that you can refer to some of my work. So dengue and other flaviviruses, what we found, like there is at the 5' UTR, they form this kind of a Y-shaped topology. Okay, so this Y-shaped topology, we found lot of pathogenic flaviviruses like dengue, Zika, West Nile virus, Japanese encephalitis virus. So those are different flaviviruses. So those, they are forming this kind of this Y-shaped topology. That apart from that, nobody has previously checked if there is some differences protein and RNA itself. So how their interacting pattern will be. So we for that we are seeing how these differences make an impact into the structural level. So apart from that, we are just checking. Am I audible?
Yes, you are audible, sir.
Okay. So apart from that, we are also checking, like what is. So for dengue, there is no molecule is there, that is pan which will inhibit. So so that was we found some of the small molecules which can be developed as a pan dengue inhibitor. That was my PhD work. So I did my PhD from BITS Pilani, KK Birla Goa campus. So after that, I moved to AIIMS New Delhi for my postdoctoral work. So in my postdoctoral, So molecular biology, I was mostly working on the SARS coronavirus too. So what in that we are doing means mostly my work is there. So I worked on like 12 variants of the SARS coronavirus too. So for that we are developing the pseudoviruses. So why to develop the pseudoviruses is that pseudoviruses are mostly replication incompetent viruses. Okay, so it is easy to study in the BSL-2 facility. Okay, so we are developing the pseudoviruses for that. Okay, it's of 12 strains, and apart from that, we are also checking like how their interactions, like how they are interacting, how the spike protein of those interact with the AS2 receptors. And not only AS2 receptor, there are other receptors also. For examples are like DPP4, APN, CCAM1. Those are some of the receptors, human receptors are there. Okay, so we are checking how these, whether they are actually forming these kind of interactions by both computationally as well as by Western blot and co-immunoprecipitations. Okay, so and entry assay also we are checking for those some of the viruses. So these are my PhD and postdoctoral work, and right now I am working on mostly these mosquito-borne viruses, mostly chikungunya virus. Right now, I am currently working similar kind of thing. Like she is also checking whether there is any significant differences at this UTR label. Okay, in the chikungunya viruses of different genotypes. Apart from that, she is mostly interested in studying this protein-protein interaction. How these, so in the protein-protein interaction, that how these viral and viral protein interaction is there. Like for example, in case of chikungunya viruses, there are four non-structural proteins are there, like NSP1, NSP2, NSP3, and NSP4. So how they are interact them among themselves, but how their interactions are there, it is not studied till now in genotypic level. So currently we are doing that. So label we are studying like how the protein-protein interaction between these viral proteins. Apart from that, we are also studying like how these proteins are interacting with these host proteins. So it is also interacting with some of the host proteins. So that is also currently we are studying. Apart from that, she is also interested to interested in the development of the small molecules against these viruses. So we are targeting different kinds of different proteins like proteases or methyl transphorases and RDRPs. Okay, for developing a small molecule drug which can be uh, that viruses. We can extend this study to other inhibitors, which we want to other viruses like dengue virus and other flavivirus, some kind of a pan inhibitor against different coronaviruses also. So and apart from that, let's see, like currently I have joined here in NIST University, so before 8 months only. So let's see like how it goes. So this is what I am and what my lab is doing. So if you have any suggestion or comment, just let me know. Am I audible?
Yes, sir. Thank you so much, sir, for sharing your research. So my one not question, just I would like to know that what is the means current status of the COVID-19 globally? Because daily we heard few news globally about the COVID-19. Hello.
Sir, yes, sir. Actually, question is not clear to me, sir.
Because now means just I want to know, sir, no, that what is the current status of COVID-19 globally?
Okay, so what is the current status of the COVID-19 globally? So currently, like I heard, like I am not right now currently into this COVID research. As I said, like I'm currently working on the chikungunya virus, but what I know, like there is some JN.1 or something, this variant is circulating and all. Omicron variant also, I think,
Omicron,
circulating and all. So I am not sure of this, but I heard of this.
because in news there are Delta and Omicron variants now. So thank you so much.
Also, Omicron variant is currently spreading and all. So JN.1 or something. So I'm not right now into this
If you have any
COVID research.
question, now the forum is open for the question and answer session. If you have any question to our Vibhu Datto Mr. Singh, you may ask now in chat box. Or you may
We
also
are causing any symptoms. So that is what this information is. But then testing is going on in the several places, right? They are making mandatory based on the purpose. The having the again RT-PCR report and all.
Sir, your question is not clear to me. I am not able to hear you actually. So that's hello.
Dr. Alami, sir.
Yeah, I was telling with reference to the present COVID testing, no, what you have asked.
Yes.
That in some places, COVID testing is mandatory based on the purpose, right? What it is there is that Omicron is actually is spreading, but it is not a very serious concern as Omicron don't cause any symptoms, specific symptoms. It is much more a milder strain of the COVID virus. That is what I just wanted to share. So you can be actually the COVID positive, but then you may not get any symptoms of the disease. That's all.
Thank you
As
so
of now.
information.
Yeah, so I want to add something. So with reference to Dr.
Alami, sir. Prakash, sir. Dr. Prakash Alami.
Alami, sir. Yeah, so actually what says said, like this Omicron variant is spreading fastly, but the pathogenic rate or like this causing disease like to that like the Delta variant. That's trying to say. Yes.
Okay. Thank you, sir. So now we would like to hear from the Dr. Sandeep Jadhav, sir, his research areas or research experience. Sandeep Jadhav, sir. Am I audible to you, Dr. Sandeep Jadhav, sir?
Hello.
Yeah. You're audible, sir.
Hello. I'm audible, sir.
Yes, sir, you are audible, sir.
So my background of research is basically on the genomics of viruses, where we bioinformatically analyze a unique type of repeat pattern, which is basically not well studied, but we want to explore that sequence type. The sequence is basically termed as mirror repeat sequence. You can understand the mirror repeat sequence like we can stand in front of mirror and we can see the image of ourselves. So the sequence is such type, and the sequence studies basically shows that these sequences are responsible for some neurological disease development, and they are responsible also for the triplexation in the DNA or RNA sequences. So in our first research, we Hello, I am audible, sir.
Okay, now you're audible.
So the first study that we come out with a virus, that is which is HIV. So in case of HIV, we find out that these sequences are thoroughly spread in the genome, and we hypothesize that these sequences are maybe responsible for maybe utilized for many perspective, like for the treatment perspective and for the diagnosis perspective, and as well as many other different kind of molecular based studies. So in our first study with the HIV virus, we find out that these sequences are actually uh, a representative of the single viral domain or single viral family. They are only found on a, for example, if we are talking about the HIV, then these sequences are only existed in the HIV viral genome only. So for verification whether whether the same sequence pattern is existed in the another viruses, we also try some other virus like dengue, Ebola, and plant viruses like TMV, and many more other viruses. So we identify there is a unique pattern of existence of these sequences in the genome of all kind of viruses. And by utilizing this fact, we predict that these sequences are actually conservative in the specific viral family, and they can be utilized for the various perspective. Our research is basically on the identification of these sequences in the viral genome. For this, we develop a bioinformatic based technique, which is a manual technique basically, and it requires 4 steps from downloading the sequence and further analysis, and its further analysis to identify these sequence type. And in all kind of our study, we find out that these sequences are basically much more important in the genome of viruses, because different and extended level of research on different type of repeat patterns are existed, like mini satellite or microsatellite DNA, and other tandem repeat, inverted repeat, and palindromic sequence. There is a very few or little research about the mirror repeat sequences. So that's why we target a mirror repeat sequence in our research, and in viral family we will find out their unique existence, and they can be utilized for many perspective, like in case of HIV, we found that these sequences are conservative, so they can be targeted for the cure perspective as well as for the diagnosis perspective also. And then we further exceed our research on the different genes, the genes at the level of genes, and in as well as in animal domain also, and on human being related genes. Our further study from virus, then I shift my study from virus to other domains, like in human insulin gene, we reported a significantly diverse mirror repeat pattern, and in case of uh, uh, maturation cycle regulatory genes, we also report the different kind of mirror repeat patterns. As well as in animal domain, we will also find out these patterns. So because of the existence of these sequences in the genome and at the different locations, it is a clear cut indication that these are significantly important. Whether it is virus or animal or plant or human being, these sequences are significantly added some advantage as well as some disadvantage also. So my basic focus currently, my basic focus is on to find out some kind of proteins that are in significantly interact with these sequence. in the cell. For example, in any virus where this conservative type of mirror sequence is existed, and any kind of protein molecule which is significantly bind with that sequence, that protein can be utilized for the treatment or cure perspective also. So my current study is focused from genomics to the proteomics background, so that we can find some proteins or enzymes that are basically bind with these mirror repeat sequences, and they can be utilized further for the cure and diagnosis perspective. So that these are the things that my lab is doing currently, and in further in my future context, I am adding some point regarding the significant role of these sequences. In the previous study, I in our previous study we find out that these sequences are also the part of regulatory unit. For example, they are exist in the initiatory regions of the trans initiatory regions of the replication or transcription regulatory part, and they are also maybe playing a vital role there for the interaction of the DNA or RNA polymerase with the genomic DNA fragment, and these kind of things we are try to find out in our further studies. So this is all. This is all from my side. Sorry. This is all from my side currently. Thank you so much.
Thank you,
If you
sir.
have any question, you can welcome.
So if you have any question, you may write in the chat box or you can unmute yourself. So there is one question from Misa Sainikita. I think that this question is for Bibudato Mishra, sir. Sir, how can next generation sequencing can be used to study the evolution of chikungunya and dengue virus lineages?
Okay, so we can there, so we can study about that. So in that way, we can think of something like how these amino acid differences are there, how the differences are there. On that basis only, like I think I have not much into this evolution of these viruses. So, but I think in by these studying these different genotype and different serotypes. Currently, I have heard like there is another serotype is also coming for this dengue. Like I recently heard, like dengue 5 is also coming. Okay, so these are mostly these how these virus evolutions are there. Like till my knowledge, like there is NH1 sequence of dengue. So if we can study these regions, so we can mostly know about their evolution. Like that's what my take is.
Okay, thank you, sir. So if there any question you can write, because we have the several experts in virology here. So anyone who want to share their experience in virology, any participants, you have even the, sir, if we have interest in working on zoonotic viruses, how can we approach any professor? So there is one question, sir. If anyone can answer this. In Naga Sri Vidya Mansuni.
So to answer your question is So what you can suppose if you are a MSc student, so if you have finished your MSc, so what you can do, you can write a cover letter and a CV, okay, and if you have any publication regarding that also you can attach, and you can search who are the faculties that they are working on this field, zoonotic viruses or any other viruses. So you can mail it to them so that they will be able to know that if they have any vacancy in their lab, or they can take you. Or what you can do, you can just appear for different national level examination like CSR net is there, UGC net is there. So if you can qualify all that, so you can, after that you can select a particular lab, those who are working on these viruses. For example, like in Odisha, like I knew some of the people, those who are working is like from ILS. One person is working, her name is Dr. Professor Soma Chattopadhyay. She is also working on chikungunya and dengue viruses. Apart from that, any other viruses that you want to select, you can just search who are the faculties members, those who are working on these areas, and you can mail it to them. If they have any vacancy in their lab, they will take you. So that's my take.
So I think that this tip is not a specific only for the genetic viruses. You can apply in any field. Thank you so much, sir. Sir, one question is I want to know about the tetravalent vaccine for dengue. Hello. So there is one question I want to know.
Is this question to me or like
Tetravalent vaccine for dengue.
is this question towards me or
Yes,
some
sir.
other? Okay, actually, sorry to say that I am not much into the vaccine research, so maybe some other faculties, those who are working on these vaccines, they may be a better person to. I am mostly into the antivirals, so they may be a better person to answer these questions.
So dear participants, just I shared on feedback form. Kindly take a few minutes and fill out it because it will help us to also issue your e-certificate and to gather the means your information. That will help us to exchange the information to the participant. Any participants, if you want to share something about the virology, now you can share it. Sir, we may conclude now, sir. Hello. So now there is no any participant want to share something.
Yes, you can continue.
Okay, sure, sir. Thank you, sir. So first of all, I would like to extend my thanks to all the participants, means all the faculty member, teachers, students, all the research scholars who have joined and supported our effort that the roundtable discussion. So I genuinely appreciate your time and involvement in this discussion. So this platform is meant to bring the researchers, teachers, students to create a space or the ambience where we can learn from each other and to grow together. And I believe that this type of the discussion is the heart of research that
जो आहे
by
त्याला
this
फेडपच झालोय मी ते जरा काढूनच टाका जाऊ दे पुन्हा
by
त्यांनी
this
एक SC लिहिल्यासारखं
and
लिहिलं. मला
last
ह्याचा प्रेसिडेंट करा, मला त्याचं प्रेसिडेंट
point
करा,
also
तुम्ही याच्यावर
mentioned
मला काढून टाकले,
through the
तुम्हाला
discussion.
फेडत झालात का तुम्ही माझ्या ऍक्टिव्हिटीला, आणि तुम्ही चेंज करता डिसिजन सतत, त्याच्यामुळं
And we also assured that we will do our best to popularize this initiative across the institution and among our young generations. And we want this to grow into a vibrant research exchange forum, one that empower the knowledge, collaborations, and innovation. So thank you once again to everyone who made this possible. And I hope that all of you will also join our next session on the next Tuesday. So thank you everyone for joining this discussion. So just take 1 or 2 minutes and fill out this form.
Okay. So I want to also thank all the organizers that they have conducted this wonderful meeting so that we have a brief discussion about this virology. So thank you all, and I am also thankful to all the listeners and all the students that they have participated. They are very enthusiastic about this topic. So maybe some of the students will pursue virology field in their future. So good luck to them. Okay. So thank you all. Thank you.
Thank you so much, sir.
Thank you.
So now I would like to request our All India President, Dr. Arvind Deshmukh sir, to share a few words with us. Am I audible to you, sir?
Hello.
Hello. Yes, Kanman.
Good evening and thank to all of you.
Thank you.
Sir, I'm looking for a PhD in microbiology. If there is any vacancy available, so please you can inform me.
So in this session,
Currently, I'm working as a
Gautam sir has shared one tip na, that how can we apply in the different institution for the PhD.
yeah.
So follow this tip, that will help. Definitely that will help you.
Currently, I'm working at Alana as a microbiologist. So I'm searching part-time PhD microbiology for suitable for my job as well as my study.
मी वैताग आणला त्याने, भलता वैताग आणला. मी आज मी इरिटंट उगच झालो, पण झालो इरिटंट. मला लिहिलं तुम्ही हर्ट करतात लोकांना.
Hello.
Yes, sir. Hello.
You are audible.
Yes, sir.
So I think that you have to search for the institution who means provide that means facility for the part-time PhD.
Okay.
And most of the institutions 6 month coursework is mandatory. So you have to be present during that 6 month at least, and must be the attendance is 75%.
Okay, sir.
You have to search for the institution which are still means providing the PhD in part-time.
Yeah.
Okay, today's discussion was very nice. Many teachers have participated today in the discussion. Bipran sir
Yes, sir.
will maintain the list of good speaker today.
Sure,
Will
sir.
upload website so that in any seminar there is a need of speaker from virology. They will call them, as well as any help from virology. They will call them. That is the beauty of this discussion, that we are getting resource person in different topics. Correct?
Yes, sir.
So thank you, Bipran sir. And next time, which will be the topic, that will decide tomorrow. Okay?
Sure.
Thank you. Thank you.
So just take 2 minutes and fill out the form, feedback form. After filling a form, you can leave the session.
Vipraj Kumar sir.
Yes, sir.
Sir, please send all information regarding PhD on my WhatsApp number. I have shared on in chat box.
You send one hi message to my WhatsApp number.
Okay, sir. Please give me your number. Taga, taga. Taga, taga, taga.